Hello friends, welcome to SAM Biology class. An electron transport chain is a series of protein complexes and other molecules that transfer electrons from electron donor to acceptor via redox reactions and couples this electron transfer to the transfer of protons across a membrane. Today we shall discuss about the mitochondrial electron transport chain, also known as the respiratory chain. All components of the respiratory chain are located in the inner mitochondrial membrane and they are concerned with the transfer of electrons from NADH and succinate to molecular oxygen. The electron carriers of the respiratory chain are organized into four membrane-embedded supramolecular complexes that can be physically separated by the gentle treatment of the inner mitochondrial membrane with detergent. Each complex catalyzes electron transfer through a portion of the respiratory chain. Out of four, complex 1 and complex 2 catalyzes the transfer of electrons to ubiquinone from two different electron donors such as NADH in complex 1 and succinate in complex 2. And complex 3 transfer electrons from reduced ubiquinone to cytochrome C, and finally it is the complex 4 which complete the sequence by transferring electrons from cytochrome C to molecular oxygen. Now let us know more about the electron carriers and the pathway of electron transport. Complex 1, also known as NADH dehydrogenase or NADH ubiquinone oxidoreductase, is a large enzyme which is composed of 42 different polypeptide chains including an FMN containing flavoproteins and at least 6 iron sulfur centers. Complex 1 is concerned with the transfer of electrons from NADH to ubiquinone. As a result of this, ubiquinone gets completely reduced to ubiquinol. Complex 1 is L-shaped, where one arm of the L is in the membrane and the other arm extends into the mitochondrial matrix. This complex catalyzes two simultaneous, obligately coupled processes. First one is the exergonic transfer to ubiquinone of a hydride ion from NADH and a proton from the matrix. So the reaction is like this: NADH plus H plus react with ubiquinone gives rise to the reduced form, that is ubiquinol plus NAD plus. So this is the function of complex 1. This is the function of complex 1. And the other process is the endergonic transfer of 4 protons from matrix to the intermembrane space. The pathway of electron transport in complex 1 from NADH to FMN and from reduced FMN to iron sulfur center and then to iron sulfur proteins and finally to ubiquinone. So the function of complex 1 is to reduce ubiquinone to ubiquinol. Simultaneously, it also pumps 4 protons from mitochondrial matrix to the intermembrane space. In addition to this, complex 2, also known as succinate dehydrogenase, the only membrane-bound enzyme in the citric acid cycle, transfer electrons from succinate to ubiquinone, because of which succinate is oxidized to fumarate and at the same time ubiquinone is reduced to ubiquinol. Compared to complex 1, complex 2 is small. It contains 5 prosthetic groups of 2 types and 4 different protein subunits. Subunits C and D are integral membrane proteins. They contain a heme group, heme B, and a binding site for ubiquinone. In contrast to this, subunit A and B extend into the matrix. They contains iron sulfur center, a bound FAD and a binding site for the substrate succinate. The electron transfer catalyzed by complex 2 can be written like this: succinate react with ubiquinone gives rise to fumarate and ubiquinol. So the function of complex 2 is to transfer electron from succinate to ubiquinol. The pathway of electron transfer in complex 2 is from succinate to FAD, from FAD to iron sulfur center and next to ubiquinol. Ubiquinol, the fully reduced form of ubiquinone, then diffuses in the inner mitochondrial membrane from complex 1 to complex 3, where it is oxidized to ubiquinone in a process which is coupled to the outward transport of protons from matrix to the intermembrane space. Other substrates such as glycerol phosphate and fatty acyl coenzyme A also donates electrons into the respiratory chain at the level of ubiquinone, but not through complex 2. The next respiratory complex of the chain is complex 3, also called cytochrome BC1 complex or ubiquinol cytochrome C oxidoreductase. Complex 3 couples the transfer of electrons from ubiquinol to cytochrome C with the outward translocation of protons from matrix to the intermembrane space. This complex is a dimer of identical monomers. Each monomer is composed of 11 different subunits. The functional core consists of one molecule of cytochrome B with 2 hemes, Bl and Bh, Reyes iron sulfur protein and one molecule of cytochrome C1. Per pair of electrons passing through the complex 3, 4 protons are translocated from matrix to the intermembrane space. This can be explained by a model known as Q cycle or quinone cycle, which accommodates the switch between 2 electrons donor ubiquinone and single electron donor, that is cytochrome. The net equation of the redox reaction of the Q cycle is like this: UQH2, this is the ubiquinol, plus 2 molecules of cytochrome C1 oxidized plus 2 protons in the presence of in the presence of complex 3 gives rise to ubiquinone plus 2 molecules of cytochrome C are reduced plus 4 protons. The pathway of electron transfer through this segment of the respiratory chain is rather complicated. However, the net result of the transfer is very simple. That is, ubiquinol is oxidized to ubiquinone and 2 molecules of cytochrome C are reduced. Cytochrome C is a soluble protein in the intermembrane space. After its heme has received an electron from ubiquinol through complex 3, it moves to the complex 4 where it donates its electron to a binuclear copper center. In the final step of the respiratory chain, complex 4, also called cytochrome oxidase, catalyzes the transfer of electrons from cytochrome C to molecular oxygen, thereby reducing it into water. This electron transfer is also associated with the outward transport of protons from matrix to the intermembrane space. Complex 4 in the inner mitochondrial membrane is a large enzyme. It is composed of 13 different subunits. Comparison of mitochondrial and bacterial enzymes shows that 3 subunits are critical to the function. Mitochondrial subunit 2 contains 2 copper ions, copper A, that are complexed with the sulfhydryl group of 2 cysteine residues in a binuclear center. Subunit 1 contains 2 heme groups, heme A and heme A3, and another copper ion, that is copper B. Heme A3 and copper ion B form a second binuclear center that accept electrons from heme A and transfer it to molecular oxygen that remains bound to heme A3. Electron transfer through complex 4 is from cytochrome C to copper A center and then to heme A and next to heme A3, copper B center and finally to molecular oxygen. For every 4 electrons passing through, complex 4 also consumes 4 protons from matrix side in order to reduce 1 molecule of oxygen to 2 molecules of water. Friends, this is all about the mitochondrial electron transport chain. For more updates, subscribe my YouTube channel, Sam Biology Class.